everyone, welcome to another lecture of drug delivery principles and engineering. In the last class we had talked about biomedical polymers and their properties. We talked about synthetic versus natural. So, what are synthetics? Synthetics is something that we are making, and natural is something that we derive from some natural form in the nature. Then we talked about various types of properties. What is the desirable properties of biomedical polymers, their mechanical, their chemical, their degradation, all those things we discussed here. Then we talked about biocompatibility. So, whether the polymer is biocompatible or not, whether it elicits immune response, whether the blood over it lyses or whether the blood is stable on it, whether the proteins adsorb to it, all those things come under biocompatibility, which we will talk further down in the course as well. Then we also talked about biodegradability, whether the polymer we are choosing needs to degrade or it does not need to degrade, again depends on applications. Whether the degradation is bulk erosion or the surface erosion, essentially meaning that if it is a bulk erosion, then you have the whole device is essentially disintegrating into smaller pieces and again randomly degrading, versus if you have a surface eroding device, then it will eventually hold its shape and just kind of degrades from the surface. So, so we discussed all of that. So, now we are going to move more into the degradation. Now, this was all degradation we talked about in terms of hydrolytic degradation. The host can also cause degradation of these surfaces. So, host induced hydrolytic processes. So, again first of all, host contains lots and lots of water. Our body is almost 90% water. So, simple hydrolysis always happens in the body. So, polymers like PGA, PLA, PLGA, all of these will degrade because the water is present and it is going to act on their side chains. You can have ion catalysis. Our body contains lots and lots of ions, including phosphate, calcium, magnesium, sodium. So, all of these ions can actually catalyze these hydrolytic reactions. So, typically polyesters, which are one of the class of polymers with the ester bond in the backbone, they will have hydrolysis happening through these catalyses mediated by these ions. So, this will essentially increase the rate. You can have local pH changes. So, of course, what is the pH of the local environment will essentially also affect how fast or how slow these things degrade. So, depending on what the polymer is, some may have a higher degradation at lower pH, some may have a lower degradation at higher pH. So, all of these will be kind of monitored. And then of course, the body contains lots of proteases, elastases and other enzymes. These enzymes are specially designed so that they can degrade whatever their target is, and they can also act on your polymers, whether natural or synthetic, since most of these degradation backbones contain polyesters and polyamides. So, those things can also cause the host induced hydrolytic processes. And then host also has capability to do oxidative degradation. So, that basically means that it kind of generates some free radicals which then oxidize the polymer. Some of the ones that degrade by this processes are polyethers, one of the major class of that is PEG, or polyamides, which are present again throughout our body as the proteins. All proteins are polyamides. So, the host induced, this could be again, as I said, it could be host induced. So, you have immune cells which are activated, such as macrophages and neutrophils. They will directly secrete these hydrogen peroxides or superoxide anion, which are strong oxidizing agents, very powerful oxidants, and they will, in presence of these, you will have a faster degradation rate than you will do in vitro if you just put it in a water sample. And this could be environment mediated. So, this could be metal ions, they can induce some cracks in your polymer device and things like that. So, all of that is also feasible. So, how would you measure bioerosion? So, the one way to do it is use some kind of an animal model, for example a mouse or a rat, and what you will do is you will place your implant into these organisms at the site that you are trying to test it. So, maybe it might be under the skin or it might be in the blood, and then essentially you will sacrifice the animal at different time points. You will take your implant out and see how much of your implant is remaining. So, that is going to give you some idea as to how fast it is degrading over time. So, then yeah, you can then essentially just measure the polymer change, the mass of it. You can see how the molecular weight is decreased or how the mass of the device is decreased, and you can do some histology to see what cells are surrounding that, how is the morphology of the device, and all of that can be done. So that's one of the way to measure bioerosion. So in the next few minutes, we are going to talk about some of these polymers that are very widely used, especially for biomedical applications. This is going to be a big list of lots of things. So I'm going to introduce each of these different classes, and we'll give you some slides which will contain essentially some of the applications for which they are used in the body. This is again essentially for your information. I don't expect you to remember all of this. Some of the common ones you'll anyways remember as we go through the course because they'll be used again and again. But don't get too worried about seeing a lot of text on the slide. This is essentially just for your information so that you have it for any kind of future reference. So first thing I'm going to talk about is polyesters. And so what are polyesters? Essentially that contains an ester bond in the backbone. So essentially these backbones are going to extend in these direction, and then this particular group is an ester bond. So any polymers that contain this in their backbone are polyesters. So they are polymers with ester linkages. Essentially, as we discussed earlier briefly, these ester bonds they have faster hydrolysis. And if they are made from, let's say, a hydrophilic polymer, then they will experience bulk degradation just because they will degrade very quickly. So some of the most common synthetic process to make them is using a ring opening polymerization. You put a catalyst and you put some small monomers with rings in them, and that essentially leads to their polymerization via the ring opening. So this is an example. So the first one, all of these three are very widely used. The first one is a polymer called PCl. Essentially you use a monomer which is ring based, and you have some kind of a catalyst, and that will essentially cause a polymerization to happen. And if you look closely, so this essentially has a O here and a C double bond O. So these are nothing but these are the ester bonds as we just discussed earlier. So these are polyesters. Another very widely used polymer is PGA and PLA. And then there's also, you can combine these two and you can also get PLGA, again very widely used polyester, one of the most widely used in fact. And again the same thing happens, there is a catalyst present here, and then that causes the polymerization to happen, and you get ester bonds which will then hydrolytically degrade. So again, as I just said, so some of the common examples are PLA, PGA and PLGA. It is also FDA approved, so it's actually being used in humans in lots and lots of devices, and we'll give some examples as we go along in the course. Very widely used for delivering molecules such as drug or gene, although they are also used for tissue engineering scaffolds. So what happens is when that ester bond breaks, it breaks into the PLA or the PGA will essentially break into the individual components such as lactic acid and glycolic acid. And which can then be ultimately metabolized by the body to produce CO2 and water. So that's how their elimination happens from the body. However, what happens is, since we have acid being secreted, you can have a situation where locally if it's a big implant, then lots and lots of acid is being developed, and if the acid is being developed, it causes a drop in the pH that may cause inflammation and irritation to happen. So you have to basically ensure that not a huge device or not a very faster degrading polymer is being put in, because that may cause inflammation and may not be biocompatible at that point. So again, as I said, degradation is fast. However, it will technically degrade by bulk degradation. However, depending on the side chains being used, if they are hydrophobic, then you can reduce the bulk degradation and shift it more towards surface degradation. this hydrolysis is also catalyzed by acids and bases, so and ions. So, if you typically the rate you will see for their degradation in water is going to be much slower than what you will see in the body, where you have all these ions and all these acids and bases that are present. So, just an example of how the hydrophobicity will matter. So, PLA hydrolysis is slower than PGA, and the reason for that is PLA contains a extra methyl group which the PGA does not have. So, that extra methyl groups makes it hydrophobic, and that is why the water penetration into a PLA scaffold is slower compared to let us say a PGA, and so their hydrolysis will be different. And then PLGA, which is a mixed copolymer of these, its hydrolysis is going to depend on composition, but it is going to be mostly slower than PGA and faster than PLA, again depends on the composition that you are using. Then you can of course also see which one is crystalline, which one is not, those things will also vary the degradation rate. Typically the polymers with the acid end groups are going to hydrolyze faster. Why? Because now you have acid present in the polymer itself, and so it can self catalyze the hydrolysis through acid end group catalysis. So, these are all little things that add up and make big changes in the properties, and you can use these small tools and changes to kind of tune your system for whatever application you are looking to accomplish. The next class we are going to talk about is polyanhydrides, and essentially this is an anhydride group. Of course, there is polymer chains here and here. This is typically formed by combination of 2 carboxyls give rise to this anhydride group. And typically the anhydrides you will find they are surface eroding, they are typically hydrophobic, and so they prevent water to prevent inside the device, and only the water can access the surface of it. Plus, as I said earlier, the anhydrides are very faster degrading functional group. So, even before the water can actually penetrate in the device, you will see that the device in contact with the water is gone, and so that is why these are typically surface eroding polymers. Such polymers will typically produce near zero order release kinetics. So, we talked about this earlier in the course. So, what is zero order release kinetics? That means that whatever is releasing from the implant is just constant over time. It is not going to change with the concentration, and if you assume that there is a big enough implant, at least for the first few time points, you will find that the thing that has come out is very similar over to the next time point. Of course, this is I am talking about very little time point. Over a long period of time, the zero order kinetics will go away, but initially you will find that they are all zero order kinetics. The degradation rate again can be controlled by the polymer composition. So, you can change the hydrophobicity, you can add more aliphatic anhydrides that is going to increase the hydrophobicity. So, you can make them degrade even slower, so they will degrade over days, or if you even make it aromatic, which is even further hydrophobic, it may take years. So, these are some of the tools you can play around with. They are extremely biocompatible. They have been actually used in clinical trials for brain cancer patient to release the drugs. So, in general the compatibility is not an issue in these cases either. Again, as I said, here is a whole lot of laundry list of different polyanhydrides that are being used for biomedical applications. You can have this slide as a reference. You do not need to remember the names of these. And then again, here is the different applications they have been used for localized drug delivery carriers. Okay, various types of drugs are being used, various kind of drug delivery systems, whether it is particles or whether it is implants, whether it is injectable. All different things are used with all different kinds of polyanhydride polymers for different diseases. So, I am just going to give you some more synthetic polymers. I am just going to run through these slides. Essentially, this is just to give you some idea of how widely these biopolymers are being used in the current literature as well as in clinics. So, some of them are polyethylene, it is very similar to the plastic that you guys use to go to supermarkets. polypropylene, PVC, polyvinyl alcohol, all of these, and their notes are written on the side. These are different applications. Again, I don't really want to go into details or any of these. These slides are here for your reference, so that in later on if you need to refer to any of these, you can come back to this. So some more of these are vinyl based, essentially CC bond. You can have polyacrylates, which are used quite a lot for light based application. You can polymerize them with light. You have polyethylene glycol, very widely used polymer, and we are going to talk about it in the next couple of classes as well. There are some other synthetic polymers for controlled drug delivery. Of course, PGA we talked about, PCL we talked about. So all of these are very widely used. The copolymer PLGA, one of the most widely used polymers out there. So all of these are there. Then you have other applications, polyurethanes used quite a lot for making blood vessels, and there are azopolymers. So different functional groups have different properties and different applications. You have silicone based implants used for breast implants, phosphorus based implants, and quite a bit used in terms of bone applications. So all of these are there. Let's quickly talk about their sterilization and storage. So now, of course, if you're going to want to implant these materials into a human body or into a live animal, you want to make sure that these are sterile, because none of your application is going to work if you have some kind of bacteria or fungus or virus present in your system. So how should you store them over long term? How should you sterilize them over long term to prevent all these complications from happening? So you of course need to minimize some premature polymer degradation. So you probably want to store it in a dry conditions. You don't want it to have too much of water present, which can then degrade it. So some air sealed samples, some moisture resistant banking, low temperature storage. So if you reduce the temperature, the degradation rate is going to go down. So that's why you see most of these fancier injections and drugs are actually stored in fridge and freezers. You have to also consider what's the hydrolysis that's going to happen in fabrication and processing of this thing, which is going to take certain amount of time depending on how complex the reaction is. So all that should be taken into account. And then once you have done this, before you use for biomedical polymer, there are various sterilization ways. Of course, heat is something that is very commonly used, but a lot of the time these polymers may not be able to sustain the temperature which are very high above boiling point and some things like that. Some other common methods are to use gamma irradiation or to expose to ethylene oxide. So essentially all of these processes are going to affect your polymer. So it comes down to choosing the lesser of the two evils. So of course you want to kill everything in terms of pathogens there, but you don't want your device to get kind of breakdown, or maybe is not able to withstand those kinds of exposures. So something like gamma radiation can significantly degrade the polymer backbone, especially the polyesters. The same thing with ethylene oxide, it's an oxidizing agent, it's also highly toxic. So if it's present if a residual amount is left, it can cause toxicity in the body. So this is essentially a big problem for the field, and lots and lots of effort has went into solving this problem. One of the other solutions are you basically make it such that it's sterile. So clean rooms have come up. These are rooms which are extremely clean. The air that is coming into the room is filtered. So if there is no pathogen present in the air, then you won't have pathogen in your sample either at the time of synthesis. Or you can filter your solutions to basically make sure anything of a certain size is cleared out. So all of these again, as I said, are strategies to ensure that your sample is sterile and is safe for storage. Okay, so we are going to talk about some of the commercial therapeutics and how these polymers will now be used to kind of enhance their effect. So more and more proteins, DNA and other biomolecules have been now becoming excellent drugs, just because they are very effective and very specific. They are highly evolved. So an example here is insulin, which is a protein. It helps in regulating the blood glucose level. So let's say if a person has diabetes, you would like to deliver insulin, but the problem is the diabetes is a chronic disease, and it's very painful for a person to continuously eat tablets or continuously get injections, not really feasible. Similarly, other examples is interferon alpha. This is used for treatment of chronic hepatitis in adults. So, it is again a small protein that is given, which is interferon alpha, that kinds of alleviate some of the symptoms that you see with hepatitis C. So, however, another problem here is these biomolecules, first of all they rapidly degrade in the body. So, of course, if I take an insulin injection today, by tomorrow I will not have any of that insulin that I had taken through the injection in my system. So, next time I eat it, I will have to take it again, because these can get degrade as well as get excreted. So, I have to take several of these to get a sustained effect, and we already talked about the dynamics that essentially for every drug there is a therapeutic level and a toxic level. And we always want to be within this range and for as long as possible. So, right now if I take this tablet and injection, I am getting a kinetics like this, however, I would like the kinetics to be more like this. So, how are these polymers are going to be used for this? So, one example is polymer-drug conjugates. So, very widely used. So, what it is you have a polymer backbone, you take your drug and attach it onto the polymer backbone, and once you then inject it in the body, what will happen is the water will come in or the enzyme will come in and slowly degrade these chemical bonds that you found at the backbone and release the drug, and the drug will continue to release out in the system till you have this drug attached here. And so, what will happen is even though you have injected the same amount that you were injecting earlier just as a free drug, the drug that is available for the system is lower. So, this drug is not going to reach the toxic levels, and then because it is going to take time for it to come out, what will happen is this is going to, instead of only remaining in the system for 1 hour, it is going to remain in the system for let us say 1 day, or depends on how big the polymer is and how much are you injecting. So, the primary advantage here is you have a very high drug loading. However, one problem here, a disadvantage here is now you have attached a new polymer to it. So, it is a new entity, it is a new chemical structure now. So, you have to get a separate approval for this, you have to first make sure that this is compatible and it is going to work in the system. The other way is, if let us say the drug is big and the polymers are small. So, let us say you are trying to inject a big protein molecule, but the problem is this protein gets degraded once in the body through action of several proteases that are present in the body. So, what you can do is you can attach some hydrophilic polymers around it, for example let us say PEG, and then what will happen is whatever the big enzyme that wants to come and degrade this wants to come, it can't come because this polymer chain repels it. So, that way you will have a lot more stability of this protein. And not only that, you have now increased the size of the protein. Earlier the protein size was only this much, now you have increased the size to this much. So, maybe now you have increased the circulation time, because remember the kidney is going to clear anything which is small very rapidly. As you increase in size, the kidney struggles to clear them out from your system. So, what are some of the advantages here? So, one of the advantages, again, you can also use drugs that are not soluble. So, let us say if your drug was not soluble, you can't inject it in the blood. However, now that you put some hydrophilic polymer on it, the solubility has improved. So, now you can inject a lot more drug without worrying about the drug precipitating. You can make it more stable just because now the degradation enzymes are not able to act on it. You have increased the half life because now the kidney is not able to clear it because it is larger. You can also modify, you can use very extremely compatible polymers to reduce the immunogenicity and toxicity. So, you have increased the safety, and not only that, you can actually on those polymer chains you can put an antibody against your target. So, that will make it more specific to it. So, it gives you more room to play around on where you want this to grow. So, let us say if I only want to target endothelial cells, I can put an antibody that binds to endothelial cells, and so that would mean that most of my drug ends up closer to the endothelial cells. And then of course, you have made it controlled release. So, instead of all the drug being available immediately, the drug is slowly getting released out, as we just discussed earlier. some of the drug will come out as more and more bonds are going to degrade. So, you have now made a control release system, and you can also make it stimuli responsive depending on what enzymes you are using. If these are only present in the disease site, then these will only release at the disease site. Okay, so what are the limitations of such a polymer drug conjugate system? So, first of all, you can lose the activity. Now, let us say if this was a protein you were going to use and here is an active site on it, but now you have conjugated a polymer here and here, this active site can no longer act on whatever it needs to act. So, you have now you have a risk of losing your activity. So, for that you need to basically be very careful in what chemistry and sites you are reacting it at, that is important. You can now you have to worry about the purification, because now you have done a reaction. So, you do not want the free polymer and the free drug to be present in your system. So, you got to devise some way to kind of clear those free systems out, and so there is another process that now gets involved. Now, the sterilization becomes a two body problem, right. Earlier you were only worried about sterilization of the drug, now you are also worried about how to sterilize the polymer. Maybe the drug can sustain a certain temperature and the polymer cannot, so now you cannot heat it. So, you have to figure out some other way to sterilize the system. And again, as I said, typically once you have now chemically conjugated this is a new molecule for regulations. So, first of all you have to test its safety, go to the regulatory agency to make sure that it is safe, and only then it can be used in the market. So, it is not like you can just change something and just directly use it in humans. You then have to go through an approval process, which can sometimes take years and has quite a lot of high cost. Okay, so we will stop here. We'll carry further in the next class as to what are the different polymers that can be used for such polymer drug conjugates. So, thank you.